use notations. Right. This is M. Chandrashekhar, Assistant Professor in Botany, MBS Government Degree College, Mehboob Nagar. In this video, we will be discussing one of the type study belongs to the class Cyanophaceae, that is a Nostoc. So if you see the classification here, it belongs to the class Cyanophaceae. They are also known as Mixophyceae. The common name is blue-green algae, right? So the class is a Cyanophaceae or Mixophyceae. Next order is a Nostocales. Family Nostocaceae, genus is a Nostoc. It is a blue-green algae. It is included in the class Cyanophaceae. It's a filamentous colonial algae. The next, it is one thing. Let me wait for a minute. Yes. Occurrence, that is a habitat. Nostoc is found in both the terrestrial and aquatic habitats. That means it can be seen, it can live in the soil and in water bodies. The terrestrial species grow as gelatinous patches on moist soils, rocks, etc. They often swell up and glisten after rains. Therefore, they are known as fallen stars. The aquatic species are mostly freshwater forms. Some species have a symbiotic association with the fungus, that is a fungi, in the lichens. See, the lichens, it is a special type of organisms. It consisting of two different groups of organisms, right? So one component will be algae, and another component of organisms is fungus. These are the fungi. Okay. So the lichens, it is nothing but it is an association. It is an association of both algae and fungi. It is considered as a symbiotic association between the two different organisms. Among these two, one will be a autotroph, and number two, that is the fungus, will be a heterotroph. They mutually help each other. Right? So that means this nostoc actually generally seen in the lichens, and in some species like Anthoceros species, like this Anthoceros is a bryophyte where these organisms, that is a nostoc, can live as a symbiont. Okay, so the association is a symbiosis where the bryophyte will help the algae, whereas the algae, it helps the bryophyte. So in lichens, the fungi helps the algae, and the algae helps the fungus. Right? We all know these nostoc actually they are considered as blue-green algae. Okay, so they are considered as they are prokaryotes among algae. So it is a special class which is there in the that is a subdivision algae. Right? They belongs to a division and Thallophyta. Why? Because the body is not differentiated into root, shoot, stem, and leaf. Okay. Next, the common Indian species are Nostoc endophytum, Nostoc verrucosum, and Nostoc commune. So these are the three different Indian species generally seen in our country. Next, it is a structure of a Nostoc colony. So this is a structure of a Nostoc colony. So if you observe the Nostoc colony, there are a number of Nostoc thalli. So these are the thalli. They are almost chain-like structures. They are known as trichomes, right? So there are, it is surrounded by a gelatinous material that is known as a mucilage. Okay. So here in this colony, consisting of more than two different individuals of Nostoc individuals. So it is surrounded by a gelatinous matrix, and the outermost region of this gelatinous matrix is known as what? It is a belly. pellicle. Right? So the structure of Nostoc colony, Nostoc is a blue-green algae. It is included in the class Cyanophaceae. They are aquatic. Most of them are freshwater forms. A few live in moist soils and rocks. Nostoc is a prokaryote. It is a colony. It is a filamentous algae. A colony composed of numerous contorted and convoluted. It is a convoluted means they are almost as a spherical like structure. Okay, so convoluted filament called trichomes. Trichome means they are hair-like structure. Since each individual, each thallus, each filament is look like a hair-like structure. That is why the entire filament is known as what? Trichome. Each trichome is surrounded by. Okay, so each trichome here it is surrounded by a mucilaginous sheath. All the filaments are surrounded by a common tough gelatinous matrix. Okay, so if we observe the each filament, we will come into the detail the structure of each individual filament. This filament is surrounded by what? A gelatinous material, that is a mucilaginous sheath. Okay, so each individual is surrounded by a mucilaginous sheath, and the entire colony, it is all the individuals, they are embedded in a general, a common tough gelatinous matrix. Due to this, the colony appears as a gelatinous patch in water or on moist soil. Because of this reason, it is often known as a star jelly, witch's butter, or Mars egg. These are the different names generally with which it can be referred. The central region of this the colony remains hollow. So the most of the portion, that is the central portion, it is a hollow. The outermost firm gelatinous layer, it is called as a pellicle. So this is a pellicle. Sometimes a few Nostoc colonies remains inside a common mucilage envelope. Such a colony is termed as a compound colony. So within this colony, if it is some a daughter colony, a separate colony will be exist. In that case, it can be considered as a a different that is a compound colony. Okay, in that case, it can be considered as a compound colony. If you observe clearly, each filament is unbranched trichome. Okay, so it is unbranched. The filament is uniseriate. That means all the individual cells in this filament, they are arranged in a single row. Okay, they are looking like a chain. Okay, so it's like a pearl beads. Okay, pearl bead. The beads are arranged one after another. So uniseriate arrangement of cells. It is made up of a linear row of cells. The cells are oval in shape, and the cells are prokaryotic. Why prokaryotic? We will see again when we are discussing the about the individual filament. Okay, some cells at regular intervals are large in size. So these are the some of the cells, they are at regular intervals. Okay, so in each and every filament we observe, there are some large cells are there. They are thick walled. So if you observe this filament here, the two cells are somewhat large and they are thick walled. These are known as what? Heterocysts. So these heterocysts are different from these vegetative cells. Okay, so these heterocysts are specialized cells. They are specialized in their function, specialized in terms of their function. So what is their function? They helps in the nitrogen fixation. That means they fix the atmospheric nitrogen. Okay, so that is why it is the main reason. Since they acting as azotrophs, since they are nitrogen fixers, they existing as the symbionts. They existing as a symbiotic association with some of the bryophytes. Okay, so these are the heterocysts, because of which they generally seen in the symbiotic association. Right. And if you observe these heterocysts, they may be intercalary or terminal in position. Okay, so these are the heterocysts. They are intercalary. Some of the heterocysts, they are almost terminal in position. So in the next diagram, we will see that thing. One second. So if we observe the individual cell, so this is the individual cell. Sorry, individual filament, individual organism. Right. So here these are the heterocysts. So these are different two types of heterocysts. One is the intercalary, that means they are present between the vegetative cells. Okay, so here in this diagram, you will be finding the two heterocysts are there. Uh, these two heterocysts are intercalary in position, whereas one heterocyst here it is a terminal in position. That is why it is known as a terminal heterocyst. Okay, so these are the two different. So the heterocysts which are present between between the vegetative cells are known as what? Intercalary heterocysts. Right. So these heterocysts are specialized in nitrogen fixation, and these heterocysts are actually they are having the pores on either side, especially the intercalary heterocysts. They are having the pores between the in the adjacent in the two terminal sites. Okay, so they are nothing but they are the protoplasmic connections between the vegetative cell and the heterocyst. Okay, so this intercalary heterocyst is having the two pores. So with these two pores, there is a connection exists between the two. That is one vegetative cell and the heterocyst. So the protoplasmic connections will be established. So here, because of the existence of protoplasmic connections, the food material can be transferred from the vegetative cell into these heterocysts. Okay, whereas in terminal heterocysts, it is having only one pore. Okay, so which will be communicating with the adjacent vegetative cell. Okay, and the cells and all the cells between the two heterocysts, they are known as what? They are known as harmogones. Okay, so these are the cells known as the harmogones. So the harmogones are nothing but it is a small filament consisting of more than 3 cells, 3 to 4, 5, whatever. Okay, more than 2 cells. Okay, more than 2 cells. The intercalary heterocysts divide the filament into two small segments called harmogones. The young intercalary heterocyst has two pores at each end. The protoplasms of adjacent cells contact with the heterocyst through these pores. The terminal heterocyst has only one pore. When it matures, the pores are closed by button-like plugs called polar nodules. So the once it is matured, okay, so the polar nodules they plug the pores so that the communication between the vegetative and the heterocyst will be getting stopped. Nitrogen fixation takes place inside the heterocyst. The heterocyst occurs mostly singly, but in some cases they occurs in chain. Okay. So generally, the heterocysts they occur singly. Okay, in the very rare cases, actually the heterocysts they present like a chain-like structure. Okay, if you observe the cell structure, the cells of a nostoc is considered to be a prokaryote. It's a prokaryotic. It is a round or oval in shape. The cells are bounded by a thick three-layered cell wall. The inner to the cell wall is the plasma membrane. It is surrounding the protoplasm, right? The protoplasm is differentiated into two regions, namely the outer pigmented chromoplasm and the inner colorless centroplasm. Okay, so we have already discussed all these points when in the general characteristic in the topic we have covered. That is a general characteristic features of what cyanophacy. Okay, so the pigment granules, cyanophase and starch, it is nothing but it is a reserve food material. Thylakoids, etc., are found only in the chromoplasm. So chromoplasm is a colored cytoplasm. It is a right. So that actually the protoplasm is divided into central central centroplasm and peripheral chromoplasm. Why chromoplasm? Why? Because it is colored in nature. Okay, the peripheral portion of the protoplasm is colored in nature. Why? Because it possessing the thylakoids. Upon thylakoids, there are so many pigments like colored pigments and photosynthetic pigments are there. So that is why the outermost the outermost protoplasm is known as what? Chromoplasm. Right? And see, the reserve food material of the nostoc is a cyanophasian starch. Okay. So actually, we are considering the cell as a prokaryotic in nature. Why? Because number one thing is that it is having the prokaryotic cell properties. one thing is that it lacks almost all membrane bound cell organelles. Okay, so there is a no membrane bound cell organelle, single membrane bound, double membrane bound. That means the mitochondria, chloroplast, that is a plast. These are double membrane cell organelles generally seen in the eukaryotes. Or these organelles, these cell organelles altogether, they are absent. Even the endomembrane system, that is a single membrane bound cell organelles like endoplasmic reticulum, Golgi apparatus. Okay, so vacuole, lysosomes, altogether they are absent. Okay, so that is why it is considered as a prokaryotic in nature. Moreover, the thylakoids are actually, they are the part of the chloroplast, the cell organelle that is a chloroplast, right? So the thylakoids are single membraned structures generally seen in the generally seen in the chloroplast. Whereas here in this nostoc, the thylakoids are not there in the chloroplast. They are simply lying in the cytoplasm. So the thylakoids are almost, they are naked. Okay. So in eukaryotes, the thylakoids generally, they are surrounded by stroma. That is a cytoplasm. That is a matrix of chloroplast. Whereas here, since the chloroplast is absent, the thylakoids are single membrane structures. They are naked. That means they are directly embedded in the cytoplasm, and these thylakoids are single membrane structures, right? And they are the places for the different pigments like a chlorophyll A, beta carotene, phycocyanin C, that is a, and the phycoerythrin C. See, the phycocyanin is a copper containing pigment. That is why it is in blue in color. That is why we call it as a blue green algae. Okay, so green coloration is attributed by the chlorophyll pigment. That is a chlorophyll A. Chlorophyll A is a chief photosynthetic pigment. Okay. So the membrane bound cell organelles are altogether absent. The genetic material, that is a DNA, is present in the centroplasm without the nuclear membrane. So this is another important prokaryotic character, right? So here, likewise that of bacteria, here in this algae, in this nostoc, a true nucleus is absent. That means the genetic material DNA is not surrounded by a double membraned nuclear membrane. Okay, so it is naked. So the genetic material is directly exposed in the cytoplasm, right? So it is there in the cytoplasm. One second. So the whatever the DNA is there, it is a naked. So it is it is generally seen in the centroplasm, that is a colorless cytoplasm, right? So the chromatin material is known as a nucleoid or incipient nucleus. So that means the true nucleus is absent in these organisms. So this is the structure, right? So the cell wall, it is almost a three-layered, and it is surrounded by a gelatinous sheet, right? So it is an individual cell. Actually, it is a filamentous form. So whereas this diagram, it is magnifying only the one of the individual, one of the cell of the filament. Okay, so generally it is like this. Okay, so this is a filamentous. So all the individual cells, that is a vegetative cells, they are arranged in a single row, right? So here it is a magnified version of one of the cell of the filament. Okay, the cell wall is a three-layered, and the cell wall is covered by a gelatinous sheet. Exactly, in bacteria also, the cell wall is surrounded by a mucilaginous sheet. Okay, that is a glycocalyx we call there. Okay, so glycocalyx. Okay, so that is a carbohydrate. So here it is a gelatinous sheet. So this is the one more similar character, which is almost similar to that of prokaryotes. Okay, so if you observe carefully, so the innermost layer, it is the plasma membrane, which is surrounding the cytoplasm. Okay, so here we don't see any cell organelles. However, only the ribosomes are there. Okay, that is a 70S type of ribosomes seen in this cytoplasm, like that of a bacterial cell, right? So that a peripheral portion is known as what, a chromoplasm. So the peripheral portion is it is a chromoplasm, and the central portion is a centroplasm, where it is enclosing a naked genetic material known as an incipient nucleus or pseudonucleus or nucleoid. Okay, it is a DNA. Okay, so in the chromoplasm, there are so many the thylakoids which are bearing the different pigments. Right, clearly we can see here. So whereas moreover the synophacin granules, they are nothing but the reserve form of food material. Also we can see in the chromoplasm. Right. Next is the reproduction. So generally, Nostoc reproduction takes place by vegetative and asexual methods. The sexual reproduction is altogether absent. Right. So in the general characters only, we have studied that the Cynophaceae, the class Cynophaceae, they don't have the sexual reproduction. So that means in the Nostoc also there is a no sexual reproduction. Okay, now we come to the vegetative reproduction. So vegetatively, Nostoc reproduces by two methods. One will be the colony fragmentation. Number two is by harmogone formation. Okay. So colony fragmentation. During the dry period, the colony becomes dry and breaks into small pieces called fragments. This process known as a fragmentation, these fragments grow into new plants. Okay, so especially during the summers, in the scarcity of water during the dry period, the colony, whatever the colony that is a gelatinous colony is there, that will be broken down into the smaller fragments. Then each fragment it can grows into a individual colony. Next is the harmogone. So harmogones are nothing but these are the vegetative cells. They are the row of vegetative cells present between the two heterocysts. Okay, so they can be between a intercalary heterocyst and a terminal heterocyst, or it may be between a two intercalary heterocysts. Okay, the vegetative part of the trichome lying between the two adjacent heterocysts is known as harmogonium or harmogone. Harmogonium is a singular, whereas a harmogone is a plural. During favorable conditions, the filament of Nostoc breaks into small pieces at the region of heterocysts. So heterocysts are nothing but they are the breaking portion, the breaking point of the filament. Okay, during the unfavorable, during the favorable conditions. Each piece is known as what harmogone. So this harmogone, where it comprises around more than three vegetative cells. Each harmogone consisting of two or more cells. The harmogone grows into filament by simple division. That is strictly speaking, it is a mitosis. It is a mitotic division, but strictly speaking, we can call it as a amitotic division. Okay, so amitosis is also known as what? Direct mitosis, where the from a single parental cell, two cells will be arised. Okay, without formation of the spindle apparatus. So where the karyokinesis is followed by cytokinesis. However, the karyokinesis is successfully done by without formation of spindle apparatus. That is why we call it as a amitotic division. Okay, so here the it is a what in this diagram we can clearly see the Nostoc harmogonium, right? So one thing. Next is so see, this is a terminal heterocyst. This is intercalary heterocyst. So this portion is a harmogone. Okay. So the harmogone, it is now it is separated from the filament. Now it is existing as what a independent individual body. Now it will undergo amitotic division. Thereby, the harmogone, the number of cells in the harmogone will be increased. Okay, so this is how actually the asexual, that is a vegetative reproduction, can be brought out. Next is a asexual reproduction. Asexual reproduction where, one second, uh, the Nostoc reproduces asexually by three methods. They are, it can be by echinite formation, or it can be heterosis, or it can be endospore formation. Number first of all, it is a echinite. Echinites are also known as resting spores or arthrospores. So some of the vegetative cells, they turn into a resting spores, especially during the unfavorable conditions. Okay. So unfavorable condition, that is maybe absence of water. Okay, so harsh conditions. The echinites are developed between the heterocysts. The vegetative cells first enlarges in size and store plenty of reserve food material. Okay, so whatever the, see, this is the vegetative cells. So before converting into echinite, it will enlarge in size. Okay, it will deposit, it will store plenty of reserve food material. So it will be used during the unfavorable condition, that to tide over the unfavorable condition, to survive the unfavorable condition, the reserve food material can be used, and it can be successfully come over the unfavorable situations. Okay, then it secretes a thick wall around it, and the thick wall is called echinite. Okay, now the echinite is nothing but it is a thick walled enlarged vegetative cell with a plenty of reserve food material. They are perennating bodies. Okay, they are perennating, that means they can survive for longer duration to tide over the unfavorable condition, and upon reversal or receiving of what arrival of a favorable condition, now it will undergo repeated mitotic division and develops into and a new plant by germination. You can call it as germination. Okay, so upon upon arrival of favorable conditions, this akinite can germinate, okay, by the process known as what? Amitotic division. Okay, so this is the method to tide over, to come over the unfavorable circumstances. Okay. Next is a heterocyst. So heterocyst is the very rare mode of reproduction. Okay. So heterocysts are thick-walled cells found in the Nostoc filament. They are asexual reproductive bodies. They may be terminal or intercalary in position. Germination of heterocysts has been recorded by Gettler and Canavius in the Nostoc commune. The protoplast of the heterocyst divide into two and then into four cells. Okay, so this is a heterocyst. So here the heterocyst is having the two plugs. Okay, so in mature thing, so the two pores are what closed by the nodular plugs, the nodule plugs. Okay, now this is an independent body. So the here the protoplast is now undergone the what karyokinesis followed by cytokinesis. Okay, so next it is a two-celled condition, further undergone the one more division results into the formation of four cells. Okay, so the heterocyst is surrounded by a thick uh, surrounded by a thick wall, right? It is a common wall. So upon upon what the reversal of favorable conditions, so the thick cell wall can be, the common wall can be broken, and the heterocyst, uh, which is developed into a new filament, will be released, and it will further undergo mitotic division and grow into a independent body. That is a filament. Okay. The important functions of a heterocyst are given below. The heterocysts act as a points of a breakage of a trichome. Thus, they can help in the hormone formation. The Nostoc commune, they act as asexual spores for reproduction. They serve as a storehouses of a reserve food. They are the sites of nitrogen fixation. Okay. So the next is a endospore formation. So the endospores, they are produced inside the heterocyst. That means they are very small in size in comparison to the echinite and vegetative cells and heterocysts. The endospores are very small. Generally, the endospores are formed inside the heterocysts. Okay. So once there is what unfavorable conditions, the protoplast of heterocysts divided into small rounded structures known as what? Endospores. So this is actually the so many endospores which are developed inside the heterocysts upon what? Upon continuous division of the protoplast. Okay, so these endospores germinate into new filament. Okay, so upon reaching of the favorable conditions, the each endospore, it will develop into a individual filament. What is the conclusion then? As there is a no sexual reproduction, there is a no alternation of generation. Okay, generally, alternation of generation includes two phases of life. One will be the vegetative, one will be the reproductive phase. Number two is the vegetative phase. Okay, so whereas here the reproductive phase, that is especially the sexual reproduction, is absent. So alternation of generation is altogether absent. Okay, so this is the summary of the vegetative reproduction and asexual reproduction. Okay, so in the vegetative reproduction, the colony can be what by fragmentation. So that is a breaking of the colony, either by hormone formation. So these are the two methods by which it can perform vegetative reproduction. Just now we have seen. Whereas in asexual reproduction, it can be by akinete formation or heterocyst formation or by simple endospore formation. Next, economic importance. So before going to that, so the nostoc is a haploid plant. It reproduces vegetatively by fragmentation and hormones. Asexually, it multiplies by means of akinete, heterocyst, and endospore. There is no meiosis and gamete fusion in the life cycle. Okay, economic importance. Many species of nostoc fix the atmospheric nitrogen in the soil. Thus, they increase the soil fertility. Reclamation of alkaline soils can be done by some species of nostoc. Okay, so the excess alkalinity can be neutralized by the growth of the nostoc species in the soil. Next, nostoc commune is consumed as a vegetable in China and Japan. Okay, so this is the life cycle just we have seen. Uh, Next, the importance of heterocysts. We have covered that thing also. Okay, so importance of heterocysts, right? Okay, heterocysts acting as points of breakage of trichomes. They helps in the formation of hormones. Okay, in Nostoc commune, they act as asexual spores for reproduction. They serve as a storehouses of reserve food, and they are the sites of nitrogen fixation. Okay, so by this we can end the Nostoc. Okay, thank you then.